Hello and welcome to Module 2. Last week we did the introduction to the course and walked through the syllabus and the Canvas shell. Today we're actually going to discuss the science of genomics. So we'll discuss basic terminology, basic concepts, patterns of inheritance, and ploidy referring to the number of chromosomes within a normal cell. But first, the why and the when of genomics for nursing. This is part 1. It's very brief. It's the importance of genomics to nursing practice and a little history to give you a background. Why are we here? Why are you learning this? Why is it required in our curriculum? Well, most diseases and factors that prevent disease have a genetic or epigenetic component. Some sources go as far to say that all disease has a genetic component to it. Certainly, it's not the only piece of the puzzle. We have epigenetics, which is the combination of environment with genetic factors. But it is important to understand the genetic basis. Next, medicine is finding new ways to diagnose, treat, and prevent illness. This is what we call precision medicine, using the information that is in your individual genome to find methods of diagnosis, screening tests for genetic diseases, treatments like the CRISPR-Cas9 treatment that was approved for sickle cell anemia, and drugs that are based on your drug metabolism, and that can be tested as well on a genetic basis, and to prevent illness based on our evolving understanding of the human genome and how we can discover information from it. The third reason that it's important is that nurses are in a unique position to help patients understand information, any information really, because that is a lot of what we do, but in particular information regarding the management of health conditions, genetic testing, what are the advantages and disadvantages, what can it see and what can it not see, risks to pregnancy and childbearing for people who are worried about inherited disorders. Lastly, understanding a person's genetic risk factors help guide us to promote health more effectively. Now, in Community Health Nursing 422, you will learn that there are levels of prevention. We have primary prevention, which is to mitigate risk factors or, help people stay well before they get sick. And genetic testing can help us do that. It can help us identify risk factors and modify the lifestyle. We have secondary prevention, which is when somebody has a known health risk and we screen earlier or more aggressively for that disorder. So knowing a person's genetic history, maybe judicious use of genetic testing, can help guide us towards lifestyle modifications that lessen the risk for disease in people and make their lives better. All right, brief history. I'm going to start in the late 19th century, this is the 1800s, with Darwin. He wrote The Origin of Species and proposed that inherited traits change in response to the environment. The presence of those traits influence mating and subsequent offspring. And it leads to that phrase that we all know, survival of the fittest, or, some people might use the word natural selection. It's a little more palatable. The problem is that genetics then became the basis for social Darwinism and eugenics. And these are social theories. They are not scientific. But A, selective breeding can encourage evolution of traits such as intelligence and character. And B, that some races are superior to others, and we all know where that has led this world. And it's a good reason that when we discuss ethics with genetics, that we remember that history, because it will guide the research and the practice as genomics becomes a more important component of healthcare. Then in 1865, we had Mendel, who published his work on inheritance using the experiments with peas, and we call that Mendelian inheritance or Mendelian genetics, and he usually deals with single gene traits, not so much the complex genetic disorders that rely on several genes interplaying with the environment, but those single gene traits can be explained by Mendelian inheritance, things like sickle cell anemia or cystic fibrosis that rely on a single gene and they are inherited in a specific pattern. In 1869, Miescher discovered that the material inside the nucleus of a cell is composed of nucleic acids, and he called that material nuclein. But his work opened up the work of other scientists who then discovered more about the structure and substance of DNA. 1940s, back to the dark times. Joseph Mengele, you might know that name, was a Nazi scientist, experimented on about 1,500 sets of twins, children, very brutally, to advance this pseudoscience of eugenics and make a more fit German nation. In the process, he tortured, disfigured, and murdered thousands of children at the Auschwitz-Birkenau concentration camp. And this is also one of the reasons that we need ethical guidelines for practice in genomics. 1953, Watson and Crick proposed that DNA had a double helix structure. It looks like a twisted ladder. They did so based on the work of many other scientists who had been studying DNA all through this time period. Then in 1977, Frederick Sanger and his team sequenced the first full genome. It was a virus. The genome was smaller than the human genome, but it sparked interest in the Human Genome Project, which began in 1990 and was completed in 2004. We will learn more about that in another module, but this is just some history. Excuse me, for you. They published the entire genome sequence. They were surprised to find that there were only about 25,000 genes in the human genome. They thought there would be many more, because there are other species that have many, hundreds of thousands. Tree species, plant species, a lot of times have more complex genomes than humans. But this was revolutionary, and now we are in an era that tries to utilize this information to identify defective base sequences, genes. It helps to understand later, when we talk about the future, CRISPR and Cas9. That's your brief history. So thanks for walking through that with me. We will come back and discuss a little bit more of the biology in a moment.